What if we could flip a switch inside our own cells that just slows down aging? I know, it sounds like something straight out of science fiction. But a tiny molecule, a peptide called Epitalon, might be doing exactly that. And a brand new study has just uncovered a secret about how it works that is so much stranger and honestly way more sophisticated than anyone ever imagined. I mean, it's one of the biggest questions we all think about, right? We all feel the effects of time. But what if aging isn't this unstoppable force? What if it's actually a biological process, something we can understand and maybe, just maybe, even influence? That's a huge idea we're going to dig into today. So to even begin to understand how we could influence this clock, we first have to figure out where it is. And it's not in our brains, it's not in our hearts, it's actually wound right into the very ends of our DNA. At the end of every single one of your chromosomes, you have these things called telomeres. Now, the best way to think of them is like the little plastic tips on your shoelaces. They don't have any important genetic code themselves, but they stop our actual genes from getting all frayed and unraveling every single time a cell has to copy itself. Okay, but here's the catch. Every time a cell divides, those little protective tips get just a tiny bit shorter. And over a lifetime of cell divisions, they just wear down and down until they hit a critical point. Once they get too short, the cell basically gets a permanent stop signal. It just stops dividing. And this process, well, that's a key driver of what we call cellular aging. So this begs a huge question. What if we could stop the shortening? Or even better, what if we could reverse it? Well, research suggests something absolutely staggering. Just maintaining telomere length could potentially increase the healthy lifespan of mammals by almost a quarter. That right there is why this cellular clock is one of the hottest topics in science right now. And that brings us to the hero of our story, a specific little peptide that has shown some incredible promise in doing exactly that. This is Epitalon. It's just a short peptide, a tiny chain of amino acids that, for decades, has shown this remarkable ability in lab studies. What does it do? It helps maintain those precious telomeres, and in doing so, extends the life of our cells. For the longest time, the scientific community kind of thought they had this all figured out. They thought they knew exactly how Epitalon worked its magic. But a new investigation just revealed that the story is way more complex and, frankly, a whole lot more interesting than anyone realized. So a team of researchers decided it was time to dig a little deeper. They really wanted to see what was actually happening inside different kinds of human cells when they were exposed to Epitalon. And they set up this really fascinating comparison. On one side, they took normal, healthy human cells, the kind that make up our skin and tissues. And on the other side, they used human breast cancer cells. And this comparison, well, it would turn out to be the key to unlocking the entire mystery. All right, so the researchers treated both types of cells with Epitalon and then went to measure the most obvious thing. Did the telomeres actually get any longer? And the answer, at first, seemed to confirm exactly what they expected. But really, it was just setting the stage for a major, major surprise. And yep, the initial results were super clear. After the treatment, telomeres got longer in both the normal cells and the cancer cells. So, okay, Epitalon was definitely working. It was rebuilding those protective caps. So the next logical question was, how? Well, the obvious answer had to be this enzyme called telomerase. It's famous for basically one thing, rebuilding telomeres. Now, it's usually switched off in most of our adult cells, but it's often super active in cancer cells, which is part of what helps them divide forever. So the assumption was simple. Epitalon must just be flipping this enzyme on. And this is where the story takes a sharp left turn. In the normal cells, telomerase activity shot right up, just like they predicted. Perfect. But look at the cancer cells. Their telomerase activity, it barely budged. I mean, it didn't increase in any significant way at all. And that was a huge puzzle. This was the real heart of the mystery. If telomerase wasn't rebuilding the telomeres in the cancer cells, something else had to be doing it. The researchers knew they had just stumbled onto some kind of hidden mechanism. So this question drove the entire rest of the investigation. The telomeres were getting longer, that was a fact. But the usual suspect, telomerase, it had an alibi. The scientists had to go looking for another culprit. And that search led them here, to something called the ALT pathway. The name kind of says it all. It's an alternative way for cells to lengthen their telomeres. It's like a backup system that about 10 to 15% of cancers use to become immortal when they don't happen to use telomerase. So, could this be what Epitalon was activating? And when they measured the ALT activity, bam, they found their answer. In the normal cells, nothing. But in the cancer cells, ALT activity went through the absolute roof. This was the missing piece of the puzzle. Epitalon wasn't just flipping one switch, it was intelligently choosing a different switch depending on the cell it was in. I mean, this discovery changed everything. Epitalon wasn't some simple on switch for one enzyme. It was acting more like a master regulator, picking the right tool for the right job. This table really sums up the incredible discovery. In normal, healthy cells, Epitalon acts like a foreman, saying, okay, let's use the standard tool, and it activates telomerase. But in these cancer cells, it's like it says, nope, that tool's not right for this job, let's grab the backup, and it fires up the ALT pathway. Two totally different methods, but you get the exact same result, longer telomeres. So how on earth does it make this choice? Well, the researchers have a really compelling hypothesis. It might all come down to a protein called histone H1. Epitalon binds to it. Now here's the key. Cancer cells tend to have lower levels of this protein. The theory is that when Epitalon binds to those already low levels in cancer cells, it triggers the ALT pathway. But in normal cells, which have plenty of histone H1, that same exact interaction leads to telomerase activation instead. It's so clever. So, what's the big takeaway here? Epitalon is a remarkably smart molecule. It uses one tool, telomerase, in healthy cells, and a completely different one, the ALT pathway, in these cancer cells. And that is so crucial. It means it isn't just blindly flipping switches. It's almost like it's reading the room and adapting its strategy to the cell's specific state. And all of that leads to this really powerful conclusion from the researchers themselves. They say that because Epitalon activates the standard regulated telomerase pathway in our normal cells and not the more chaotic ALT pathway, it suggests it could be a safe way for healthy people to maintain their telomeres as they get older. You know, research like this shows we're moving way beyond just fighting against aging. We're actually learning to communicate with our cells in their own language. And it really makes you wonder, as our knowledge keeps growing, will we one day be able to fine-tune our own biology with this kind of incredible surgical precision? It's a pretty fascinating thought.